move on to our next speaker, our own Susumu Tanagawa, who will tell us about the coming of age of the memory engram.
Okay, good afternoon. How we extract information from our experience to represent this information extracted from experience as what is called the memory traces, and also how we use them subsequently for the process called memory recall or retrieval. Obviously, is one of the fundamental questions in neuroscience. So today, I have ambition to squeeze in these four related subjects. As is written here, it could be a little bit too much. Eddie already warned me that I should not go over the time. Well, luckily, Li Hui asked me to give some conclusions of this today's event at the end of the day. So if I cannot finish it and allocate the time, I can conclude. I can give my conclusion during that 15 minutes. Anyway, so it's only 100 years ago, a little bit over 100 years ago, that this idea of the memory is stored as traces in the brain network, what it's called engram, that was presented in a more formal manner by this man, Richard Zeman, German psychologist, who published a paper in the early last century, but actually English version came out about 15 years later, 1921. So, which is called the Mudeme. Okay? In this book, he elaborates the concept of memory trace or memory engrams as we know a little bit more these days. Okay? However, his idea was completely ignored by contemporary and the people who followed him until 1982, when Daniel Schachter published this book and tried to revive the original Zeman's idea. Okay? And according to his idea, which is modified based on our contemporary knowledge, the conceptually is like this. I call this engram theory, Zeman's memory engram theory. So when you form a little bit of a memory from your experience, it is postulated somewhere in the brain network, a population of a cell get activated, whatever that means, activated. And then they undergo some enduring, this is important, enduring physical or chemical changes. And in such a way that subsequently, when information which is related but can be a partial part of the original set of information stored as a memory returns, then these cells get reactivated, and that leads to what is called a memory recall or memory retrieval. Okay? So in modern days, question is, of course, what kind of changes we are talking about here? And we use all these things here, synaptic plasticity, sort of pioneered by Hebb, enhanced excitability of cell-wide excitability, immediate allergen expression, other gene activations, protein synthesis, and structural spine or synapse changes. Now, there has been a large amount of literature which support this concept during the past 100 years. However, when it comes to the entirety of Siemens engram theory, it has been very difficult to demonstrate it from the not only molecular physiological point of view, but all the way based on the animal's behavior. Okay, so I would say that the information gathered until by a few years ago was each one of them are very useful, but it's more fragmentary. For instance, the person I really respect, Sina Jocelyn in Canada, Toronto, she, some years ago, in one of the reviews, she stated a minimal requirement for proving, for demonstrating the validity of Siemens engram theory. And that is simply put this way. A direct test of memory engram theory would require specifically labeling only the neurons involved in memory formation being activated by experience, and then subsequently reinstating memory recall by reactivating those neurons, specifically reactivating those neurons. So with the advent of a variety of genetic technologies combined with the more sophisticated behavior paradigms and so on, only a few years ago, Shu and Steve attempted to come up with this more coherent or more extensive evidence for the theory. So we use contextual field conditioning, which everybody knows. So this is a context-specific Pavlovian conditioning for rodents. This is the general protocol of the approach Steve and Shu employed. You start with this transgenic animal shown in here. C-force is one of the immediate-early genes. So promoter drive the expression of tetracycline-dependent transcription factor called TTA. Okay, you start with transgenic mouse, and you inject this into the, in this case, hippocampus, and we chose the gyrus for this work. In fact, with the virus, which TRE is basically the promoter to which TTA can bind, and which drive the expression of channel rhodopsin EYFP, and inject them into try to target this to the gyrus. Another thing targeted is the optic fiber. Okay, you grow this animal in the presence of doxycycline until the day of experiment, and then you switch the two things. One, a diet is now doesn't have any doxycycline. Secondly, the context is switched from A to B. Okay, and then you gave a training, in this case, mild foot shock, and this will allow, if C-force protein gene is activated by this training, then this will allow the labeling of the potential engram cells with the channel rhodopsin. Okay? And then the next day, you put it back to the context A, not the context in which shock was given, but a different context. And the animal will not freeze because the context is too different. Two contexts are too different. But the big question is when you activate the engram cell, would the animal freeze? Can you induce the artificially by activating the putative engram bearing cells in the context in which animal was not shocked? So answer is yes. And unfortunately, I don't have this one working. So here. Okay. So this is a busy slide, but focus on this one here. So this is a testing day, right, off, on, and this is a freezing. And you can see here the reversible freezing by these animals, only when when the light is on, when the Q30 engram is activated. So this satisfied the requirement Sina described in that review. In other words, these are likely to be what is called, what Zeman originally proposed as engram cells. In this case, it's a contextual engram cell. Engram cells for encoding context, specific context, in which a shock was, normally the shock is given in contextual field conditioning. Now, memory is usually quite reliable, as we all know. However, under certain conditions, it's been known in humans that people, apparently normal people, make a very gross error in forming memory. And this has been called a false memory. It's not an unsure memory. It is a very sure memory for the experience the person could not have encountered. Okay? So I'm not going into the detail, but after DNA test was introduced, there has been a survey of the criminals who have been imprisoned many years by serious crime, and they test the DNA, the criminal's DNA and DNA kept in crime sites were compared. And according to this survey, 3 out of 4 people DNA sequence did not match. By the way, these were selected, these cases were selected for those in which the testimony, human testimony played a major role for the incrimination. But there has been no false memory model in animals. Okay? So we had, based on our early findings that I just described, we had this, we decided to test the following possibility. That is, a false memory could be created in animal by an association of a recalled past experience they formed before, which is kept in mind as a CS, conditioned stimulus, with a concurrent external event of high valence at the US. Okay? So CS US, in this case, doesn't happen at the same time, in a separate temporal gap between them, but they can form a memory and by associating CS and the US. Again, this is published, so I'm not going into very detail, but this is a protocol. You put the animal in A context and label the cell, but because doxycycline is not in there, and then go through this control. Animal goes through the control environment, C, which is all these environment A, B, C are different from each other, and then put in the B and give a shock. However, at the same time, A context engram is activated artificially with light. Okay, and then question is subsequently, do animals show the A context engram, not only a B context engram, the peer memory, but also A context peer memory. Okay? And the answer is yes, as you can see here. They clearly, they form a memory for the context in which they never acquired shock. Okay? The others were controls. Now, this memory actually is a real memory, as you can hear. So it's kind of a contradictory statement, but real memory means that this type of memory can be reactivated later in the context which has nothing to do with the context they have animal experienced by illuminating cells, false memory engrams, as indicated in here. So that's a real memory. In fact, in the human studies, people have been having a problem distinguishing by functional MRI studies the difference in a way in the area of the brain which lights up when a person is giving you the genuine real memory versus giving obviously the false memory. So that's why these people really believe that something happened to them. The experience was there, although it cannot be. Okay? But anyway, so this is now I want to change the subject to this one. How is the variance associated with episodic memory encoded in the hippocampal amygdala circuit? Hippocampus is neutral in encoding variance of the experience. So somewhere else, the variance is encoded, but when most of the experience episode comes with some influence to your emotion. So there must be some kind of circuit interaction. And the obvious possibility is the amygdala is involved in this. So in order to ask this question, Roger and Josh did the usual thing, that is label the cell bilaterally in one set of mouse in the dentate gyrus, but another set of mouse in amygdala, VLA, okay? But again, bilaterally. And they can go through peer conditioning as well as reward conditioning. For reward conditioning, we use the male subject and then let the male play with the female for half an hour to 2 hours. That gives the reward memory to the male subject. So anyway, so the expression, the labeling is quite happened very well in dentate or VLA, regardless of whether fear memory or pleasure memory. And then they developed sort of a symmetrical the recall measuring protocol, and which is based on our optogenetic reactivation recall that we found before. So you put the animal in this rectangular box, in one box. I can just show you the other way around. So each side has some kind of visual cues. So animal went through fear conditioning or pleasure conditioning, and you put the animal in here, and they can walk around through this box as they want. But when after those animals which you went through the fear conditioning, you put in this box, and then each animal have some kind of preference initially for the one with the two zones, two sides. So for the fear memory recall, you put the animal, and you take the staying time without any kind of further manipulation in the preferred zone, okay? And then you give, when animal move around here, you give a light to activate the fear memory, the fear memory, the trace, only when animal is on this preferred zone. Okay, you put the light there, and animal recall the fear memory, and therefore they avoid, they run away from there to the other side. Okay, so by subtracting the basal level of staying time from the staying time after light is given in that target area, you can get a negative result as the indication of fear memory recall. So for the animal which has gone through the reward conditioning, you do the reciprocal studies. What is you start with unpreferred zone at the baseline staying time, and then you give that light only in the target areas, and then the animal likes that recall the good memory, present memory, therefore they tend to stay there rather than running away. So you get a positive result if you subtract the baseline. Okay, so here is the protocol. So this is the mouse in which the animal went through the fear conditioning, and then the avoidance test was conducted. In both then the gyrus labeled mouse or BLA labeled mouse, you get indication of fear memory recall. It's a negative different score, okay, which is what we expected. If you put the animal in a paradigm of pressure memory, then you get both the gyrus labeled or BLA labeled animals show the recall of pressure memory. The big important question, interesting question, is what happens if you let the animal go through fear conditioning first, but then subsequently you use this inception protocol. You reactivate those engrams, but now with the US of opposite valence. So the fear to pressure here like that. Okay? But the same CS is used here and there because we are activating the original A engram with the light. Okay? So question, so if you do that with the gyrus engram, you get here the switch from negative to positive memory recall. But interestingly, you label the BLA engram, then they don't switch. They cannot switch in terms of valence. And the same reciprocal situation exists if you start with reward memory and then go to the fear memory. Okay? Only in case of the gyrus labeling, you can get a reversal. Now the interesting question is what happens when you get this reversal from start with the fear and then go to reward? What happened to the original fear memory? Well, you can find out by subjecting after reward inception, subject them to what is called the opto-place avoidance test, and which will tell you the recall of original fear memory. And then you can see here the fear memory is greatly reduced when animal go through the reward experience in between, okay, from here to there. So from this, we conclude something like this. This is our proposal. I mean, there are many details, maybe more finding is necessary, but basically, in hippocampus, let's say in this case, the dentate gyrus, not directly, but indirectly, information reaches the amygdala, BLA, and amygdala BLA cells apparently are hardwired for two types of cell, excitatory neurons. One is for storing aversive, the reward memory, and the other is for storing the fear memory. Okay, so when the US is changed from the negative to positive, what happened is, although this basal connection, anatomical connection is there for both type of BLA cells, but plasticity or strength of connection apparently get switched when the US is changed, okay? And at the cost of the connectivity of the other pathways. So this is the way we interpret our result. Now, I want to go to the last subject, which is something like this. So Steve wanted to take advantage of to find out whether this valence switch that was found by Roger and Josh and so on can be exploited to rescue or to reduce, at least to reduce the stress-induced depression type of behaviors. because obviously depression type of behavior is driven by the series of negative experiences. And as you know that in depression in the human, there are a tremendous number of people suffer this, 350 million worldwide, and then 1 million deaths per year, and a large amount of money is being spent to purchase the antidepressant. So hypothesis that Steve wanted to test is something like this. Reactivation of hippocampal cells active during the encoding of a positive experience may be sufficient to rescue stress-induced depression-related behavior. Okay? Of course, in this mouse, okay? We're not working on human. These behavior include a lack of effortless, motivated responses to challenging situations, which can be tested by what is called the tail suspension test. Okay? The second major symptom of depression is the inability to experience or seek out pleasure, which is called anhedonia, which can be also measured in case with mice with what is called the sucrose water preference. So the animal is a bit thirsty, and 2 bottles of water is given, one with the sucrose, the other not. And then normal animal would prefer the sucrose water, but the depression model of mouse does not distinguish. Anyway, so here are the first set of experiments Steve conducted. There are 3 groups of mice here. Okay? And this is a cartoon for the demo showing the protocol step by step. So this is the so-called experimental group. So animals were labeled for their pleasure experience in here, okay? Pleasure experience memory. And went through this neutral experience, but not labeled. And then animal go through what is called a chronic immobilization stress protocol. You wrap them up. And for 10 days, 15, I think 2 hours, 2 hours a day for 10 days. They hate it. But then, and then the next day you test animal for these 2 different behavior, tail suspension behavior and the sucrose preference behavior. And as the original engram is activated online with light and on and off. Okay? And this one is a control in which animal did not get any kind of stress treatment. And that one is another control in which the memory engram labeled is not a positive memory, but it's a neutral memory. Okay? And you can see here that control animal, this is the suspension time. The time when the animal will try to deal with the struggle is relatively high, independent of light illumination. Okay? But and same thing is the sucrose preference. They prefer sucrose water whether light is off or on during the testing. But experimental animal initially is low in the struggling time, but significantly goes up if you turn on the light, activating the positive memory. Okay? And so it's a sucrose preference. Initially low, but it goes up by light. And this neutral memory doesn't always show the low struggling time, and basically no preference for sucrose independent of light. So it looks like that activation online, acute activation of a positive memory engram is rescuing or reducing the depression-like phenotype. So do I have enough time to show this? Or I'm not sure. I think 5 more minutes? Yeah. Anyway, I skipped this. I mean, this is just to show the animal is struggling under the the normal animal will struggle longer compared to the animal which went through depression protocol. And also this shows that if you turn on the light and activate positive engram, then animal behave as if the one which did not go through the depression the protocol. Anyway, so what Steve further wanted to find out is to find a potential circuit which may help, which is helping the activated positive memory resulting in the rescue of depression type phenotype. And so he did this brain-wide survey of C4 positive cells during tail suspension test upon positive or neutral dentate gyrus engram reactivation in the stressed mouse. Okay, so I won't go into detail, but summarize this. And he found that the C4 activation is particularly high in the nucleus accumbens shell compared to the core, and also relatively high in amygdala, both BLA and CEA, but not very high. It's not high in medial prefrontal cortex. Okay, there are other places which showed higher C4s, but anyway, he wanted to find out whether this pathway from hippocampus to BLA and the BLA to nuclear accumbens shell might be the major pathway which is helping activated positive memory engram to rescue the stress and also the depression type phenotype. So he did this very sophisticated by brain site and by opsin. So these are 4 different combinations, channel rhodopsin and RT, and see whether this pathway really is important. Okay? So what they did is they took, so in this case, as usual, the dentate gyrus engram for the positive memory is labeled with the channel rhodopsin, but in the same mouse, also the input coming from BLA or amygdala to nucleus accumbens is also labeled with RT. Okay, and now you subject this animal to the tail suspension test, and you can see here the indicated in here that inhibition, the rescue ability of light-activated DG engram cells, positive DG engram cells, is blocked by inhibition of BLA input into the nucleus accumbens. But this is not the case if you look at, if we inhibit input coming from medial prefrontal cortex to nucleus accumbens. So there seems to be some specific role of this hippocampus, amygdala, and nucleus accumbens shell. So this is the final work. All the rescue experiment I have been talking about is acute one. Actually, it happened only during the behavior test. Okay, so it's an online rescue. Okay, so he wanted to find out whether under certain conditions this can be prolonged. This rescue period can be prolonged. So what they did is as indicated in here. Positive neutral memory is labeled and went through the stress protocol, and now animal was the positive memory was activated multiple times. Okay, 5 days, twice a day, each time 15 minutes or something like that. You write them up, and compare that with the one day lighting. And as you can see here, if you animal go through this, 5-day treatment of the positive, 5 days lightening of the positive memory engram gives significantly higher the struggling time or sucrose preference compared to the one-day treatment, one-day lightning. Okay? And also, if we look at the individual animals, there is a significant correlation between sucrose preference and time struggling in the tail suspension test. So I think I'm done. Yeah, so good. So here is the people who did made major contributions. Shu Lu is here, and Steve Ramirez and Arvind Rennie not here anymore, but he started this entire project. And this reversal of valence thing is Roger, Kim, and Autumn Aarons. And also, actually, these people have also worked on this. As I said, Steve was a major person who worked on this rescue of the stress-induced depression phenotype. These people, I'm not going to mention each one of them, but they have contributed very important ways in various aspects of all these 4 different projects. Thank you very much.
So we have time maybe for 1 or 2 questions.
Is it also possible to take, instead of pleasure, the exercise and enrichment, and then you could activate the same engram?
Do you mean to see whether exercise would help? It could, because we also find that rescue lead to more neurogenesis in a dentate gyrus. So that correlates with that. And people have claimed that exercise will induce neurogenesis in dentate gyrus, so it could be related. Yeah.
So how long does this memory could be kept? Like how long do you wait? Can you wait? It's like, can you wait for a month or a year and then test again?
Definitely not a year. Even a month, probably not. Okay. However, if you look at it like 2 weeks in a mouse, okay, it depends on the species. In mouse, other studies Roger is doing about system consolidation studies by manipulating engrams, dentate engram. It looks like in 2 weeks they are still quite active there. But a month later, no, not so much.
Does it differ between pleasant and unpleasant conditioning?
Present?
Yeah. Does it matter whether the stimulation condition is a pleasant?
By the way, if you're talking about the rescue, then the longest we have looked at only 3 days. Okay, only 3 days, not even 2 weeks. I mean, that doesn't mean that it doesn't work, but we have not looked. Yeah.
My question is, have you tested the offspring? Is the next generation could be increase the sensitivity, or?
I didn't understand.
Say offspring.
Yes, yeah.
Upstream.
Yeah, no, offspring.
The pups.
Oh, pups. Oh, I don't know. I mean,
Because there was the wrestler paper.
you think this would be inherited to the next generation?
Yeah, that's right, because the wrestler paper is showing
Some kind of epigenetic.
the olfactory.
I think Li Fei will find that out.
Okay.
Sorry, we don't know anything about it.
Any more questions? Okay, thank you, Susumu.
You're welcome.